demonstrate how to use our gene dosage maps within the UCSC Genome browser. So first, I need to demonstrate how you use the Genome browser. So here's the URL for the University of Santa Cruz Genome browser. And so here's their homepage. And on that homepage, you want to click on Genome browser. And it will take you to a second page where you want to select which genome assembly you want to look at. And in this case, you want to pick the one that says HG19. And so select that. And it will take you to a page that probably doesn't look exactly like this, but you'll see near the top an ideogram of a chromosome. And below that will be a window with a white background that has a lot of data in it. That's your viewing window. And below that, you'll see, you might have to scroll down the page, but you'll see these blue bars that go across the page. And above the blue bars, you'll see a row of tabs. And one of those is going to say Hide All. And I've got a red circle around that. So click on Hide All. And then you'll see a screen that does look more like the one I'm showing you right here. So the parts of the screen that you see here are, of course, as I said, the ideogram of the chromosome at the top. Below that is the viewing window, and below that are the blue bars, and some of them may be open and some may be closed. You can click on the plus or the minus to see and click on the plus to see what's included in each of those types of data. There are, each one of these, if you select them, is called a track, and each track has a certain type of data or from a certain source of data, and there are probably 100 of these different tracks to choose from, and they're important to genome junkies, but they're just not really important for someone with a clinical interest. So that's why I had you start with Hide All. The ones you do want to look at are circled here in red. You want to look at gene position, and so there's a little drop down, and you want to select dense. You want to look at chromosome band, and you want to select dense there. And you want to look at the UCSC genes, and in that view, you want to pick PAC from the drop down. So select those three, and then hit any one of the refresh buttons over on the right hand side. And then you'll get a screen that looks a little more like this. And so you see the chromosome ideogram with the red box around some portion of it. And the portion in the red box is then zoomed in on right below that in the white box, and the data for the three kinds of data you just selected, the genome position, the chromosome band, and the UCSC genes in the region are shown right below that in the viewing box. So those are the three things you've selected. So next we want to go to the position in the genome that you want to investigate. So that is in this box. And up here you can write, there's a couple choices of how you can approach what you want to look at. You can write a gene name or gene acronym in that box and click on refresh or click on go, and it will take you to that gene. Or you can write a chromosome band like CHR18Q23, and it will take you to that band. Or you can write the genome coordinates. In this case, you can see just to the left of the circle are the coordinates on chromosome 18. So, and the format is shown right there. So you have an example. It says CHR and the number of the chromosome, a colon, and then the base pair position. So if you click on that, it'll take you to this region of chromosome 18Q. And then you should see a screen that looks like this. So what you can do from here is select a gene. So let's just click on the t-shirt gene right here, circled in red. And if you click on that, that's where the interesting stuff starts to happen. Then it'll take you to a details page. And on this details page are lots of different kinds of data. At the top is a description of the gene, and the full gene name tells you what the t-shirt stands for. The green box is an index of what other information is on this page. But below that, in the next box, are all the databases that have information that pertain to this gene. So 2 of those have clinical information in them that could be relevant. One is PubMed, of course. It'll take you right to PubMed and all the articles about this gene in PubMed. And also OMIM, Online Mendelian Inheritance in Man, and that will explain the clinical data associated with that gene. So many of these other databases aren't going to be relevant, but these are the 2 that are most important. You can also go to the mouse genome database from here, lots of other places, and you'll get the data associated with this gene in those databases. So that's how to use the UCSC Genome Browser. Most important thing is start with hide all, so you just don't have to even look at all that other data that has no clinical relevance. And so now that you know how to do that, you can use our gene dosage maps. And the 2 reasons you might want to use our gene dosage maps for chromosome 18 is to find out information about individual genes or individual phenotypes associated with chromosome 18. But you also may want to look up the clinical consequences of a region on chromosome 18. So either way, for whichever purpose, our gene dosage maps have 2 tracks, actually. There's a gene dosage map for information on the specific genes on chromosome 18, but there's also a phenotype dosage map. So the phenotypes associated with chromosome 18, where a gene has not been identified yet, but a region of the chromosome housing the key gene has been identified. And we have color-coded our information in our gene dosage tracks. And they're in the following colors. There are 263 genes in total on chromosome 18. The ones color-coded green are considered variants. And by that, we mean, I guess, they're not associated with gene dosage issue. So could be a gene associated with a recessive condition, for example, would be color-coded green. It's not relevant for gene dosage. Those that are color-coded yellow are considered risk factors. So deletion or duplication of that gene by itself does not cause a phenotype, but it only is associated with a phenotype in conjunction with another chromosome change or gene change or environmental exposure or something, some other factor in addition to that gene. Then the pink ones are dosage sensitive genes. Red ones we think are haplolethal, and the ones color-coded gray are unknown. We have no idea if it has dosage potential or not. So those, stay tuned. And these are the numbers of genes in each of those categories so far. And you can see 198 are considered variants by us. And so that's the vast majority of genes on chromosome 18. So there's not so many that we really are concerned about. 24 are risk factors, 22 at this time are considered dosage sensitive, and 19 are completely unknown. So those are the ones we are investigating if they're in a phenotype region of interest. So this is what the gene dosage map looks like. You can see the red box, and this, now we're in the genome browser, and the red box on the chromosome ideogram tells you that the viewing window below is a zoomed in view of that region of the chromosome, and you can see all the genes in that region, and they're all color coded. Let's pick the t-shirt gene here. If you click on the little line, the pink little line of the gene, that will take you to a details page in the genome browser. And so now you've got to click on the gene name again here, and then it will take you to our database that shows you the information that we've collected on this gene related to gene dosage and the references that support that information. So if you want to look at the phenotype map, here are the phenotypes, and you can tell this is the phenotype associated with the whole chromosome, because the red box on the chromosome ideogram is around the entire chromosome. And so the viewing window shows all those phenotypes that are associated with chromosome 18. Some are in gray because those are linkage regions, for example, and so there's no mechanism known, so we don't know if it's relevant to chromosome copy number change or not. The green ones could be recessive conditions linked to the chromosome. The pink ones are the ones that we associate with a deletion or duplication of chromosome 18. And again, this is a whole chromosome view, so you can see there's quite a lot of phenotypes in which the region has been significantly narrowed. So let's pick one at the bottom, which I now realize you can't see for the video cutting off that corner, but we'll pick the bottom one here, and it just says DM. And so here's the details page in the Genome browser, which you have to click on again to get to the DM part, to get to our page. And here you can see that that's a dysmyelination, and here's the information about that and the references to support that designation. So here is the whole chromosome view, showing all the genes. Kind of hard to see them at this resolution of the whole chromosome, and the phenotypes. And this particular view just shows the ones that are dosage sensitive. And so the point here is there's lots of specific information you can find about genes or regions. If you know the region someone has deleted or duplicated, you can look that up and see what specifically is associated with that. I hope this helps you learn how to use our gene dosage maps, and keep checking back because the information is updated continuously. Thank you.